So the final stage of meiotic prophase 1 is the dikinesis and the terminalization of chiasmata in the dikinesis, and the chromosomes fully condense and the meiotic spindle is assembled to prepare the homologous chromosome for separation. Meiotic spindle is prepared, and by the end of dikinesis the nucleolus disappears and the nuclear envelope breaks down. Dikinesis represents transition to metaphase. Then there is the diplotin. The beginning of diplotin is the dissolution of synaptonomal complex and the tendency of recombinant homologous chromosome bivalent to separate from each other except at the sites of crossing overs. The X-shaped structures are called the chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. So this one is, and then there is crossing over leads to recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of packaging, leaving the chromosomes linked at the sites of crossing over. Crossing over is also an enzyme mediated process, and the enzyme called recombinase. And then there is the four chromatids of each bivalent clearly appears as tetrad, and the sites at which crossing over occurs between non-sister chromatids of homologous chromosomes. Non-sister chromatids, that is the different chromatids of two homologous chromosomes. That's the statement in here. The crossing over is the exchange of genetic material between two homologous chromosomes. Non-sister chromatids, let me mark this point. Non-sister chromatids of homologous chromosomes. Then there is bivalent or tetrad, that is synaptonomal complex form, and the first two stages of prophase 1 are relatively short-lived compared to the next stage, that is the packaging. There is a zygotine stage, that is a synaptonomal complex formation. There is a liptotin stage, that is becomes gradually visible under the light microscope, shows the liptotin. The compaction of chromosomes continues throughout liptotin. This followed by second stage of, and there is prophase 1 of meiotic division, typically longer and more complex when compared to prophase of mitosis. Leptotin, zygotin, packaging, diplotin, dikinesis.